Hello, welcome to Biology, the scientific study of life. Embryonic development involves cell division, differentiation, and morphogenesis. In the embryonic development of most organisms, a single cell zygote fertilized egg gives rise to cells of many different types, each type with a different structure and corresponding function. For example, an animal will have muscle cells that enable it to move and nerve cells that transmit signals to the muscle cells. A plant will have mesophyll cells that carry out photosynthesis and guard cells around stomata pores, which means that regulate the passage of gases into and out of leaves. Within a multicellular organism, cells of different types and organized into tissues into organs into organ system, and organ system into the whole organism. Thus, the process of embryonic development must give rise not only to cells of different types, but to higher level structures arranged in a particular way in 3 dimensions. Notice this figure, this photo illustrate the dramatic transformation of a zygote into an organism. This transformation results from 3 interrelated processes: cell division, cell differentiation, and morphogenesis. Through a succession of mitotic cell divisions, the zygote gives rise to a large number of cells. Cell division alone, however, would produce only a great ball of identical cells, nothing like an animal or a plant. During embryonic development, cells not only increase in number but also undergo cell differentiation, the process by which cells become specialized in structure and function. Moreover, the different kinds of cells are not randomly distributed, but are organized into tissues and organs. The physical processes that give an organism its shape constitute morphogenesis, meaning creation of form. Notice that. Notice A, fertilized eggs of a frog, and B, tadpole hatching from egg. Notice that. Notice also from fertilized egg to animal, what a difference a week makes. It takes just 1 week for a cell division, differentiation, and morphogenesis to transform each of the fertilized frog eggs shown A into a hatching tadpole like the one in B. Notice that a protective jelly coat surrounds the eggs and tadpole. The processes of cell division, differentiation, and morphogenesis overlap in time. Morphogenetic events lay out the basic body plan very early in embryonic development, establishing, for example, which end of an animal embryo will be the head, of which end of a plant embryo will become the roots. Notice that. These early events determine the body axis of the organism, such as the interior posterior head to tail axis and the dorsal ventral back to belly axis. Later morphogenetic events establish relative locations of structure within smaller regions of the embryo, such as the appendages on a fly's body, the fins on a fish, or the digits on a vertebrate limb, and then within regions still smaller. Cell division and differentiation play important roles in morphogenesis in all multicellular organisms, as does the appropriately timed programmed death of certain cells. However, the overall sequence of morphogenesis in animals and plants exhibit significant differences. In addition to many shared developmental mechanisms, the development of animals and plants differs in 2 major ways. First, in animals but not in plants, movement of cells and tissues are necessary to transform the early embryo into the characteristic 3-dimensional from the organism. 2. In plants but not in animals, morphogenesis and growth in overall size are not limited to embryonic and juvenile periods, but occur throughout the life of the plants. The structure responsible for the plants or for a plant's continual growth and formation of new organs are apical meristems, Preparatory embryonic regions in the types of shoots and roots in animals. Ongoing development in adults is normally restricted to the generation of cells that must be continually replenished throughout the animal's lifetime. Examples are blood cells, skin cells, and the cells lining the intestines. During differentiation and morphogenesis, embryonic cells behave and function in different ways from each other, even though all of them have arisen from the same cell, the zygote. Okay, let's talk about some case stages of development in animals and plants. Cell division, morphogenesis, and cell differentiation occur in both animals and development and plant development. Molecular events leading to cell differentiation begins as early as the two-cell stage, but observable differences among cells are not evident until much later. Let's begin with animal development. Most animals go through some variation of the blastola and gastrula stages. The blastola is the sphere of cells surrounding a fluid-filled cavity. The gastrula forms when a region of the blastola folds inward, creating a tube, a rudimentary gut. Once the animal is mature, differentiation occurs in only limited way for the replacement of damaged or lost cells. Notice that zygote fertilized eggs eat cells, and blastola cross section and gastrula cross section got here adult animal sister. Second one, plant development. Notice that this one in plants with seeds, a complete develops within the seed morphogenesis, which involves cell division and cell wall expansion rather than cell or tissue movement, occurs throughout the plant's lifetime. Apical stems parable continuous arise and develop into the various plant organs, and the plant grows to an indeterminate size. Let's begin zygote, fertilized egg, and this one two cells, and the imperial inside seed and seed leaves, and also shoot, apical meristem, and here root, apical meristems. Okay, thanks for listening.